Hello, everybody online. This is Marcus. We're going to wait a few minutes to get started. This traffic's kind of bad this morning, so we're going to let everybody roll in.
There we go. As of yesterday, the CCHA. Yeah.
Can people online hear us?
Loud and clear.
Sounds good. Yep.
Okay. If I had a gavel, I'd bang it now, but I don't have one. Welcome, everybody. Small group today, not to be unexpected. I know it's been kind of a crazy time lately, so I really appreciate folks showing up and still participating, even though it's been stressful. I'm running the meeting today. Normally Kelly and Ryan would be here, but of course they're tied up with emergency response duties, so we'll be running the show today. So this is our last TAC meeting of the year. We made it. We have a lot of good talks today, and hopefully we'll have some good time for discussions and Q&A and all that good stuff. So we got Carrie first, and then we have John Bowden from UF, Amanda and Jay talking about T-Bond, and then Ray talking about some updated modeling work. Before we get started, I think we'll just do a quick round robin of who's in the room. There's 29 people online, so we'll skip them. Sorry, online folks, but we'll just say who you are, where you're from, and on a scale of 1 to 10, how good you feeling this morning. I'm Marcus T-Bep. I co-chair the TAC. I'm running about a 7.5. Not too bad.
Regulable PSA, I'm with you on this. Could be worse.
That's right. Brandon
Johnson with Stantec.
Christine Davies, Miles County. I'd say like Stacy Day, Nellis County. My office is in shambles.
Blake Simmons, Tampa Bay Estuary program, like an 8.
Greg Honorado, Fish and Wildlife, I guess 7 also.
Matthias Kramer, FWC, FWRI. Yeah, a good 9.
Oh.
Yeah.
Casey Craig, FWRI Coastal Wetlands, and I'm saying probably about a 7. Kara Redabot, FWC, FWRI, 8.
Julie Christian, Environmental Protection Commission of Hillsborough County, about an 8.
Hi, Emma Dantes with Miles County, solid 7 today.
Sydney Lane with U.S. Fish and Wildlife Service Coastal Program. I go with the 7 as well.
Amy Butler, Tampa Electric Company, 7.
Carrie Clary Walia, Restoration Ecologists program. I'll go with an 8. Nice.
That's awesome. It's nobody below 7. We're all like solidly average. Okay. Well, first up we have Carrie, who will be talking about some work that she recently presented at the AFS meeting in Hawaii. So should be well rehearsed. Controls.
Thanks, Marcus. Good morning, everybody, and thanks for making it in person. I know that it's a struggle to make even normal things happen right now, so appreciate having you here. I'm going to be talking about some data analysis work that we've been looking at for the last couple of years, trying to link Necton communities to seagrass loss and recovery within Tampa Bay. And I'd like to acknowledge first my Estuary program co-authors, Marcus and Ed, and then also Megan previously with FWC, but she's now down at Everglades National Park. So like Marcus said, I presented this at AFS, but I'll probably gloss over some of the background that I did there since everyone in the room and online is pretty familiar with the Tampa Bay system. And a lot of the things that we've been talking about over the past couple of years are the fact that we've been seeing a lot of signs of stress in our seagrass communities within the Bay. We had a really good recovery up until about 2014, but in the last 3 mapping cycles done by the Water Management District, we've seen a pretty drastic decrease. So, just taking that as, kind of background, this is a survey that happens every couple of years. So we're looking forward to seeing the next round of mapping information that should be out shortly. But for right now, it looks like we're on a little bit of a decline when it comes to seagrass. And the majority of that actually has been happening in Old Tampa Bay. And if you look at the graphic, as you see the seagrass kind of increasing and then decreasing, a lot of that acreage has been gained and lost in that bay segment. You can actually look at this in a little bit more detail if you break it down even farther into seagrass management areas that the Water Management District has designated. So the graphic that has all the green on one side, that's the increase up until 2016. And then on the other side where you see most of the decreases in the red, you can see that a lot of those large increases in acreage and decreases in acreage, again, are happening in the Old Tampa Bay segment and specifically in a few areas like Feather Sound. So that's kind of the background on seagrass. And, we're also monitoring in conjunction with FWC, the fish communities using the Nectin Index. So we're also seeing signs of stress there. So the Nectin Index was developed based on the fisheries independent monitoring data that's collected monthly within the bay. And this index is based on the small fish communities that are inhabit the bay, that are usually associated with seagrass beds and unvegetated flats. And it's a really great data set. I worked in that program for a long time. And basically, this index was developed in between the Estuary program and FWC, and it's basically an index that's based on species richness. It's a multi-metric index, but it gives you a general idea of the health of the fish populations in the bay. And based on that analysis, we have a management framework that we use within the Estuary program to communicate how well the populations are doing over time. So if the index is in the green, we want to stay the course. The fish populations are doing well as compared to previous sampling. If it's in the yellow, we might need to do some additional research to see kind of what's going on within the community and get to the bottom of why there's some decreases. And then if it's in the red, we need to be on alert and do some management.
The almond spice.
Me too. Get one for me. So as you can see, the index has varied over time. We haven't been in the red, which is a good thing. But you can see these really low dips there. And those are associated with significant red tides that went into the bay in 2005 and 2021. So this index is really good at picking up on those large scale disturbances. But the question we want to ask is, can it help pick up and reflect some of the seagrass changes that we're seeing in the community? And, how can we go about doing that? And kind of merge some of the data sets that we look separately over time. So the objectives of this study were to evaluate the potential drivers of Nectin community patterns, whether they're spatial, whether they're within those base segment areas, whether they're in the smaller seagrass management areas, temporally by season and by year. Obviously, with fish communities, you're going to have a strong seasonal pattern depending on recruitment of juveniles into the system. And then we also want to look at, water quality, environmental drivers that might be affecting these patterns. And then in addition, the ecological patterns. So, the amount of seagrass per year, what species are associated with those seagrass communities and all those different types of things. And the end goal of this would be to kind of quantify the relationships between the Nectin and seagrass dynamics to inform management. So that we can kind of link those up instead of having two separate metrics. So this is a little bit more detailed of a breakdown of the seagrass acreage based on mapping up at the top. And this separates out the seagrass acreage by bay segment. So Old Tampa Bay on the side, Hillsborough Bay, Middle Tampa Bay, and Lower Tampa Bay. And, like I mentioned, Old Tampa Bay's been showing the steepest declines. Hillsborough Bay has a pretty low cover of seagrass to begin with, but that's also showing some declines. Middle Bay a little bit as well, but Lower Tampa Bay looks pretty steady. So what we wanted to do is take the Nectin index values and see if we can match them up to the mapping values. So the Water Management District, again, only does that assessment every couple of years. So we took the Nectin index and collapsed it and averaged it into those separate seagrass assessment years. And what you see here is that we can see a pattern of the Nectin index potentially tracking those seagrass acreage numbers. So especially in Old Tampa Bay, you can see a decline in the last couple of years. Hillsborough Bay is kind of all over the place. Middle Bay, you see a little bit of a decline. In Lower Tampa Bay, you don't see any declines in the end. You actually see some increases. So that's just looking at a really general annual part of it, but there's additional information that's taken both from the Water Management District and from the Fisheries Independent Monitoring crews on the ground. So we can get more detailed information on the quality of the seagrass beds. So for example, the Water Management District not only does acreage, but they separate them out into two different types of seagrass cover. So whether it's continuous or patchy, none. They also do algae, but for the purposes of this, I'll talk about continuous versus patchy beds. And the fisheries independent monitoring biologists, when they're out in the field on the day of sampling, they're estimating cover. They're also looking at the species composition of the seagrass within that sampling site. So they have more detailed information on whether it's a thalassia bed or a haliduli bed, things like that. So we wanted to take these two data sets and see if they kind of track each other over time. So this graph shows you on the x-axis, these are the categories that the Water Management District uses to designate different areas. And then on the y-axis is the total percent SAB cover that the biologists on the ground are measuring. And remember that this is over a couple year period where we're looking at the FIM data. So, you know, even though it looks like there's some seagrass in the none category, that could be over, you know, a couple years of sampling. But the main pattern you see is what we would hope for is that, you know, the Water Management District mapping data is matching up with what's being seen on the ground. So patchy seagrass has a higher percent cover and continuous seagrass has the highest percent cover. So we do know that the mapping data mirrors the field data, so that's good. So let's see what happens when we use the index to track this as well. And fortunately, it's as you would expect. The index increases as the coverage of seagrass increases. So this isn't rocket science, and you know this is something that would be obvious to all of us in the room, but it's a good test case for using these two data sets to kind of link them together. We can go a little bit farther though. The Nectin index is a general species richness index that doesn't really give you the full breadth of, you know, what type of species are being caught in a particular time. So, you know, even if you have, you know, 10 species caught in, you know, multiple years, it may be a different composition. So what we can do next is actually look at the different species that comprise those sampling events and see if we can glean some additional patterns. So the next thing we did was put together a multi-parameter analysis that's basically trying to look at all the different environmental drivers that could influence the fish communities. So this is a multi-dimensional scaling plot, and I'll just orient you to the symbols and the colors. So the colors are the season. So black and red are summer and fall, winter and spring are blue and green, and you can see that those are very well separated. And the symbols have to do with the base segments. So you can see up at the top, it's mostly Hillsborough Bay, which is the star, and Old Tampa Bay, which is the triangle. And then down towards the bottom, you'll see more of the middle bay and lower bay. And those vectors that are on top of this MDS kind of give you an idea of why it's separated the way it is. So if you have higher temperatures and higher water depths, that's going to be more summer and fall based assemblages. And if you look going to the bottom of the graph, there's higher percentage SAB cover, more acres of seagrass, you know, higher proportions of thalasity and salinity. So that's more of the middle bay and lower bay. So what we have here is the main differences are seasonal differences separating the communities. Secondary to that, you've got the spatial factors, whether it's the bay segment, and we did incorporate the management area as well. So that also had an effect on the seagrass communities. So this is looking at some of the environmental factors. You can also overlay species onto these MDS plots to see what kind of patterns fall out. And this is the same MDS plot. It just has species that contribute, that correlated with up to 60% of the community structure. And I wouldn't worry about too much the type of species that are in here, but you'll notice that most of them have higher abundance in the summer and fall. So, we're getting higher abundances of those seagrass associated species. And then I bolded a couple that I'm going to talk about later, the clown goby, the leatherjack, and the spotted sea trout. So in order to get more of an idea of what's going on, we wanted to subset these data to look specifically at Old Tampa Bay, where we're seeing the most differences in seagrasses. So the next slide is actually showing the species-specific trends for species that contributed up to 70% of the differences in the community. And these are average abundance over the different water management area designations for seagrass. So what I want you to notice here is not necessarily the species, but to notice that, you're getting higher abundances as you go up in seagrass cover. So lower abundance for most species with no seagrass, higher abundances with continuous seagrass. And, one of the things that we wanted to get out of this is, we have this Nectin index and it's a good way of communicating general health, but a lot of times when we're communicating that, it doesn't really resonate with the public. It's like, oh, what's Nectin? What does that mean for, when I'm going to go out and fish for red drum or whatever. So we wanted to look and see if we could find some specific indicator species that could be a reflection of those seagrass acreages. So just to point out a couple, so two of the fish that contribute a lot to differences in seagrass, that contribute a lot to the differences in community structure are Gulf pipefish and spotted sea trout. Gulf pipefish obviously is very associated with seagrass. My 7-year-old says it's because they wiggle a lot like seagrass and that's why they like to be there. And then juvenile spotted sea trout, is very much associated with seagrass in its juvenile life stages. So what we would expect, looking at abundance over time, is that we would see increases associated with seagrasses. We're not necessarily seeing that yet, but this is just kind of the first step in looking at what we can figure out as far as patterns. We're going to be doing some additional general linear modeling to get at why the patterns are the way they are. But you do see some increases like on the back end for Gulf pipefish and also within spotted sea trout, but then you see this decline at the end. So it's possible that they may be tracking that. On the other end, we wanted to look at fish that might not be as associated with seagrass cover. So we looked at clown goby and leatherjack, which if you look on the side there, there's not a huge difference in abundance over all those three categories. They're not necessarily increasing with increasing seagrass cover. And then if you look at the abundance over time, you can see the clown goby's just been increasing. And that's not super surprising. They can hang out in very muddy habitats. And then leatherjack, which is more kind of a pelagic species, that's just extremely variable over time. So the take home from all of this is basically that, our Nectin index is really good at reflecting large scale disturbances over time on an annual basis. But by going through this exercise, we found that we can match up seagrass measured in the field with mapping data pretty well. And that also the Nectin index and species specific trends may reflect changes in seagrass habitat. So this is a good first step at getting at what we want to do. And the next steps, we're going to look a little more detail in those 4 species that I mentioned, do some general linear models and look at all the different factors that are associated with those trends. And then we also want to look into incorporating the Nectin index and seagrass cover into a management framework. So for example, is there a certain threshold of seagrass we need in the bay for us to be in the green on the Nectin index? And then in the meantime, as always, we're monitoring that report card over time and continuing to report that to the community. And we're also funding riverine nectin index development that we're working with FWC on. They're putting on some additional sampling in the Hillsborough River so that all 4 rivers within Tampa Bay are being monitored. So in addition to having a bay index, which is the one I've been talking about, we'll also have a river index. And that's all I have. If you have any questions, be glad to answer them. And if you're like me and my brain doesn't work, this month, that's fine too.
Out of curiosity, have you guys started looking at all with the benthic index as well and seeing if that matches up at all with either the nectin or the seagrass loss?
We haven't, but that benthic index is something that Marcus and I have talked about is definitely underutilized. And, we'd love to talk later about how we can use that to better communicate the conditions in the sediments. So.
Yeah, the challenge with all of our report cards and indicators is they're all very modular, and we don't necessarily do a good job of linking them together to understand is, for example, association of seagrass with connected. One would expect there to be an association, as was clearly demonstrated, but it's not always this like a one-to-one relationship. And we are actively trying to figure out the best ways to relate them, particularly for management outcomes, as Estuary mentioned. And yes, the benthic index is pretty underutilized. It's another line of evidence that we want to get a better handle on. And it's right for this type of analysis. There's just so many pieces of information that could be telling how does it affect benthic species.
Yeah, and there might be a lot more interest in that type of work now with, all the concerns after the hurricanes and what's been going into the system and, looking at some of the historical contaminants that are already being measured. So. Yeah.
So I know a lot of the seagrass losses in Old Tampa Bay are linked to the low flushing and poor water quality up there. But what about the losses in Middle Tampa Bay? What are the theories behind those declines?
It's a good question. Could be a lot of things.
We've been talking about this for a long time, ever since it started declining from 2016. And yeah, the elephant in the room is Old Tampa Bay, like you said, related to flushing and production of sediment, that sort of thing. If you look at the loss of seagrass in 2016, it's not just limited to Old Tampa Bay or Tampa Bay in general. All along the southwest coast was declined seagrass. So it's likely related to a regional phenomenon, and climate change could be an issue there, whether it's temperature stress or lots of variation, precipitation, particularly more precipitation over time. And I think the fact that Old Tampa Bay and Middle Tampa Bay, and to some extent Hillsborough Bay, because they're higher up in the more towards the receiving end of the watershed, they're more influenced by these large regional effects than say Lower Tampa Bay. So that's what I think is going on, but it's still, there's no clear, this is what caused it. It's just a concern that we're all worried about and still trying to figure out what's driving it.
It's too many manatees.
For the record, TBEP does not advocate calling it manatee.
Well, thanks, everybody.
Hey, Carrie, I had a quick question. I'm trying to find my little hand raising thing, but I don't know where it is. So I'm just going to speak up. Hi. Hey, just a really quick question. So first of all, I'm really happy to see that these data are lining up. That gives me a really good confidence in using these indices. But I'm wondering, do you guys get any perspective from either the commercial fishing or like the charter boat captains, as far as like looking at maybe more vicarious species that might grab the attention of people more than nectin or seagrass? Do you kind of have an idea of how these numbers jive with kind of what they're seeing out there every day?
I mean, I think that's kind of what we're trying to get at. So the Nectin Index was based off the FWC Fisheries Independent Monitoring data, and really the only data set that could be used reliably was the small seine data, which is the very small individuals. So what we can do is we can look at juveniles that are caught in those gears. So for example, like the spotted sea trout. So we could potentially look at that. FWC does a really good job at doing single species assessments, and they're actually going to regional management right now for a lot of those more popular species. For example, snook and red drum are now under regional management. And within that, they look not only at the indices, but they look at the incidences of red tide. They look at the seagrass declines and or increases. And then they also look at, what the fishermen are thinking over time. So that's actually is being incorporated in some management through FWC. And I don't know if anyone else wants to chime in on that in the room, but so I think it's being looked at, and we kind of want to get to the point where we can look at not just a, individual index number. Again, Nectin doesn't really mean much to, the general public, but, if we can find one particular species that they're interested, that we can track that over time, I think that's where we're headed.
Okay, cool. Thank you. Yeah, it sounds great.
So there are no more questions for Carrie. I just will follow up to that to say that, the TAC is a place for us to share ideas. And as she expressed, this is a work in progress. Do you have any specific questions of where you think this could go for the group? Or is there any guidance you would like from people that are either in the room or online?
Yeah, I think what we're struggling with, and Marcus has done kind of some initial playing around with kind of how to incorporate this into a management framework. So, if there are other species maybe that we want to use, or if there's a different way we want to look at the Nectin Index that would communicate these challenges better, we're definitely open to it. So, like I said, reach out and give us some ideas.
Stay tuned.
Okay, thanks.
So next we have Dr. John Bowden from UF. I need to make him a presenter.
Good morning, everybody. Can you hear me?
Yes, we can.
Okay, good. Wish I could be there. Unfortunately, I'm at a conference this week. I'll let you guess which state I'm in by the decor in the background. But and I think today I'm probably about a 7.5 myself.
Hold on, I'm gonna do it on my. Do you have it now, John?
Yes.
Yes, yeah, so Benny from Texas, is that right? You're at a conference right now?
Yes.
Hence the longhorn in the background?
Yes. Huge longhorn in the background. So.
Cool.
You may need to stop sharing your.
Oh, yeah, I need to stop sharing it there. All right, there we go. I think.
Is the slide on the screen?
Yeah, it's like screen and screen, but I think we're good.
Okay. All right. Well, good morning. My name is John Bowden. I'm an associate professor. I guess I didn't update my slide. I'm an associate professor at University of Florida, and I'm going to talk a little bit about the PFAS work we're doing across the state of Florida, and then talk specifically what's happening with what we're doing in Tampa Bay, and then also talk about some of the things that we're doing now and also planning to do. And so let's see if I can move my slide. All right, so our lab is based on using novel chemical techniques to try to assess where emerging chemicals of concern end up in our environment. So we're really interested in developing new workflows to catch up with all the new chemicals that we need to monitor. When you look at their fate and transport, where they end up in food chains, once we throw these products away, where do they go, how they get back into the environment, and then we also look at their toxicity. We're funded by a lot of different agencies. I like to put my acknowledgement slides up first, so all the students and all the funding agencies get their proper due. And so far, right now, we've been working on PFAS for about the last 8 years. We have about 60 publications working on PFAS, and we're very collaborative. So just briefly, I'll talk about what our lab is all about, and then I'll talk about a little bit about PFAS for those of you who don't know much about PFAS. We're a lab that's based on developing new chemical tools, and you'll see that as we go through the presentation, to keep tracking these chemicals once they get into our environment. And our lab has the complete orthogonality of chemical detection, so our instruments are able to collectively look at just about anything that ends up in our environmental samples. We're really big about education and awareness. So we train students from our lab and other labs that come in. We teach them how to do PFAS analysis, and we try to be vocal educators about the PFAS problem. We are very collaborative, and we have a really big interest in community interaction. So we, as you'll see in our data sets, we actively recruit samplers, and we go in to a lot of different workshops and community engagements to talk about PFAS and water quality. And we're also big about data transparency. So everything we do, we make sure is available to everybody and they understand it. So why are PFAS an issue? These are called forever chemicals, and that's because they're made up a bunch of these carbon-fluorine bonds, which is one of the strongest bonds in chemistry. They also have this kind of duality of chemical structure. So they have this hydrophilic head group, which kind of likes being in water, and then his hydrophobic tail, which doesn't like to be in water. So that really gives them a lot of unique qualities that are used in a lot of industrial and consumer products. Unfortunately, as a society, we a long time ago, we said, hey, we want to buy cheap products, but we also want them to last a long time. And the crazy chemists got in the lab and they made these PFAS chemicals. So, unfortunately, those same properties that they last a long time and they have this duality of chemical properties make them a really big challenge to track and get rid of in our environment. Unfortunately, these chemicals are in just about everything. I think the last number that I saw, there's close to 15,000 of these chemicals. And, I'm not really that surprised today about them being found in just about every kind of consumer product you can think of. Probably most notably is use of aqueous film forming foams, which are used to put out fires at military bases and airports. And of course, Florida being a large swamp, once those are sprayed, eventually there's runoff and there's storms, and those chemicals get into our environment. I won't spend a lot of time on this slide, but I really do like it because it really puts in focus a lot of the major contributors to PFAS into our environment. So you have manufacturing effluent, you have landfills, you have use of biosolids on farms, you have use of these AFFFs on airports and military bases, and of course there's wastewater treatment plants that don't always properly remove PFAS when they have their effluent. And, of course, we in Florida, we rely on the water, we rely on wildlife and tourism, and of course, some places for our drinking water. And so really tracking how these PFAS get into our environment, how do we get rid of them, are really key things that we should be focusing on. And so our lab, we have a couple different assays. We have a targeted assay, which basically means that we have a known set of PFAS we're trying to monitor. This is kind of what the EPA does. We also have a non-targeted assay, which I'll talk about a little later, which is where we try to uncover what those unknown PFAS are that are in our environment. And then there's a whole bunch of new kind of frontiers that are kind of now the new thing people are looking into for PFAS, which are looking at the true volatile PFAS, which are not captured with typical targeted methods today. And what we're finding is that a lot of these true volatile PFAS that are emitted in our indoor spaces, that are emitted at landfills and industrial sites, are actually just as concentrated or higher in concentration, and we know virtually nothing about their toxicity. So that's something that really should be paying attention to. And then also I'll talk a little bit about total fluorine later and why that's important. These are a bunch of different matrices that we've published and worked on, and so you can see PFAS get everywhere, and looking at them in all these different matrices just tells us a little bit about their story. We look at them in environmental samples, a lot of samples that are based on waste products or are part of our waste. Some of them are beneficial reused, so it's really important to track if there's PFAS in them or not. We also have a lot of matrices that tell us about our exposure levels, and then also looking at their kind of integration into the environment. If you don't know, PFAS has been on EPA initiative since 2009. It's kind of taken them a little bit of time to get kind of cranking up, but you might be aware of the 2016 drinking water guidance values of 70 parts per trillion. That's for PFOA and PFOS. 70 parts per trillion is like a drop of water in an Olympic-sized swimming pool, so it's a very low concentration. But it wasn't really until recently in 2023 that these proposed maximum contaminant levels are enacted. So within several years, at least for 2 PFAS, they're actually going to have limits that you have to be below is 4 parts per trillion. But as I mentioned, there's 15,000 of these, so we still have.
Miller Construction.
Okay.
They only submitted the.
Right. Anyways, so what kind of data do we have in Florida? So there's this really great page, if you haven't seen it, through the Environmental Working Group that shows you some of the PFAS data. It looks like there's a lot of data in Florida, but Florida is actually quite big, and there's really not a lot of data points. And so part of our research is to try to fix that. And so I'm going to briefly talk about some of the projects, and most of everything I'm going to show you is published. And so if anybody wants any of the publications, I'd be happy to share. One thing that we really kind of wanted to do was what we call crowdsourcing sample collection. So we get public interests involved, and then we have the public help us collect samples across the state. Obviously, the state of Florida is huge, and so this has really helped us generate some really comprehensive data. So we got people who are interested in collecting drinking water, and we started this project in late 2022. We sampled 450 taps, and we came up with this kit. We shipped all the boxes out, and we had a SOP protocol on how to collect the water samples. And on the right, we have a heat map. And so these heat maps are generated by each tap that's monitored, its concentration, and then its relationship to other points or other taps measured. And so what it creates is this heat map that tells us kind of approximately where we should be focusing on for future efforts looking at drinking water contamination. And this is total PFAS. This is the sum of all the PFAS we measure in our targeted assay. And you can see here that, by and large, some areas are doing pretty well and some areas are having some issues. Tampa Bay actually wasn't one of our hotspots for drinking water. There's one little red point down there, but for the most part, it was pretty good in terms of in comparison to the rest of the state. But I'd be happy to share more data if anybody wants to look at that. We also did another paper looking at PFAS in Florida Springs. And so, I had an undergraduate student that was like, do we see PFAS in springs? And I was like, let's do it. So we sampled 50 springs across the state of Florida. And sure enough, we find PFAS in springs. And interestingly enough, we collected samples from both the vent and the run. And almost in most of the sites we tested, we didn't see a difference between the two. Which is kind of concerning to us, but also something that we're hoping to explore further. But to get to the big project, we use the same crowdsourcing technique to try to map out PFAS across the state of Florida. And so this started in 2020. And the idea was we wanted to create baselines that were good for prospective monitoring, but also to identify hotspots. Again, we had a kit. We have a YouTube video that shows people how to collect the samples. And overall, we had over 130 undergraduate students help collect samples. And we also had a lot of engaged citizen scientists and organizations help collect. And we had over 2,300 sites that we monitored. So this is a quick a picture of the total measured PFAS. Again, this is the sum of all the PFAS at each site. And you can see as the points get redder, the higher the concentrations are. We looked at 92 PFAS in our method. 33 were detected in at least one sample across the state. The highest was 3,000 parts per trillion, which is insanely high. And our average was 29 parts per trillion. So this is a really cool thing, right? But what we wanted to do was use these points again to create another heat map, because that'll tell us about where the hotspots are in Florida. And before we get to the heat map, I just want to point out that I mentioned before the maximum contaminant levels in drinking water was 4 parts per trillion. And so we use that as a basis to assess what the levels are in the surface water. And on the left, you see for PFOA, we had in the state of Florida over 900 sites that were above that MCL. And basically anything that's kind of middle yellow or closer to red is a site that's above 4 parts per trillion. And on the right side, we have for PFOS about the same. And so you might see there's a little table here with the number of samples above that 4 parts per trillion. Then PFOA in Pinellas and Hillsborough County, we had quite a few, the 2 highest of all the sites counties we looked at. And Pinellas was among the highest for PFOS as well. So definitely kind of triggered us to maybe we need to start paying more attention to Tampa Bay. This is the heat map I told you about. So taking all those points, we were able to generate a heat map that kind of told us where in the state are our PFAS hotspots. Again, on this left, this is total PFAS, so this is the sum. And then we also can do this for individual PFAS. So PFOA, this is basically the hotspots, and anything that's kind of orange and redder is above that MCL. If you kind of blow up the Tampa Bay region, you can actually look at, again, the sum PFAS, but also a bunch of individual PFAS from your short chain PFBSs to some of your long chains like PFNA. And for the most part, it's pretty red in the Tampa Bay area. There's some differences across the PFAS, but that's some of the work that we're trying to get to in our next round of studies. So once we had all this data, we really wanted to kind of see what this kind of meant. And I kind of went into this thinking that AFFFs, the use of those aqueous film forming foams, was probably one of the main sources. I happen to know about this spills database by the Florida Department of Environmental Protection. Since 2017, they've been tracking the number of spills in the state of Florida, and you can actually access this online. And during the time of the sampling, or prior to sampling, there was over 7,000 spills with 1.4 billion gallons of a bunch of different things emptied into the waterways. And so we actually went through and tracked all those and put all those spill locations on a map. And so on the right here, you can see the spill locations. And if you were to just look at that and kind of, here's an example. If you were to look at those spill locations and compare that to the heat map on the left for PFAS, you see a lot of similarities. And so this really got our attention that perhaps some of these spills, especially of course, like during hurricanes, may be large contributors to PFAS. And because PFAS don't degrade, they don't take a long time to break down, this could potentially be a significant source. So we broke down this into different quadrants. C is the Tampa Bay area. And as you can see here, there's quite a few spills. And again, these dots represent a different spill. We can blow this up, and we organize the spill types. So this is going to be looking at raw sewage. And then we wanted to look at not just location, but the amount that was spilled. And so here you can see this is about, in the dark orange, so about 6 million gallons of raw sewage over that time period was spilled into the Tampa Bay. And we could also do this with untreated wastewater. Of course, these values are much higher. These are in the bottom. There's 100 millions of gallons, and you can see here that we had kind of in the middle range around 9 million gallons or so of untreated wastewater spilled into the Pinellas area, and a little bit higher that's in the Hillsborough area. So what we did with that is, once we knew how much was released, we looked in literature to see how much is typically found in these types of waste matrices. And we were able to calculate how much PFAS was potentially released in grams. And this we just did for 2 PFAS, so PFOA and PFOS. And you can see here that over the course of that 2017 to 2022, we had about maybe 60 grams estimated emptied into Florida waterways, and for PFOS, much higher. And what's really interesting here is that in many of these matrices, so let's say like wastewater, you can see here the influent and the effluent. You can see that not much of the PFAS is removed during most of these processes. So we're just kind of dumping this stuff right into the waterway. So again, I'm all about data transparency. So I'll say that we're building a research website. So if you go to the page, the link's up top there. Many pages aren't finished yet. Our release date for having everything completed is 11/15, but you can go to the webpage right now and you can click on the PFAS research, go to Florida surface water data, click on that, and you click on whatever county you want to look on. So if you want to click on Pinellas County, you can pull it up right now. It has a Google map that you can kind of zoom in on. It shows you all the points. And then below that, all those points that have an ID, you can actually track and see what the concentrations are that we found in those samples. And you can, if you want to zoom in, if you can't see, you can look at individual PFAS and then the sum of the PFAS in each of these sites. So that's available right now that you can look at. We're hoping to make this a little bit more interactive than we have right now. Doing web pages is not my forte, so we're kind of painstakingly getting there. But this data and all the data from other projects are going to be online as well. So that's what we've done. And so just for a few minutes, I'm going to talk about what we're getting into. And Tampa Bay, because of its high levels and some of the data we have, is one of our test sites. And we've identified several locations based on data, based on potential PFAS sources that we're going to start tracking. And just as an example of the state of PFAS measurement, right now we're doing the targeted assay, which looks at a handful of these PFAS. But the reality is we really have no idea the totality of PFAS that we're exposed to or that are in our environment. So the couple assays that we're doing, that we're working on right now, that we're hoping to apply to Tampa Bay, I'm going to talk about here in a second. So there was a really cool paper that came out recently looking at surface water in South Carolina. And what they did was they looked at the targeted method, which I've been talking about, and they did something called total organic fluorine. So if you were to take a sample and you were to break all the fluorines off of everything in that sample and measure the fluorines, that's the total fluorine, right? So that tells you the potential capacity of all the different things that are in there. So if you think about our targeted assay, obviously some of those compounds contribute to that total fluorine. But as was shown in this study, which you look at the orange bars versus the blue bars, the orange bars were massively higher than the blue bars. And what that tells us from this study is that we're only in these South Carolina waters, we're only measuring a small fraction of all the PFAS compounds that are actually present in these environmental samples. And this got us to thinking that, all right, if we go back to some place that we know has a lot of PFAS in concentration based on our previous data, can we do that same analysis? Can we see how much we're actually monitoring of the total PFAS that are there? And so we have all the extracts of all the samples that we collected for our first study, but we're actually going out again this year and maybe most of next year to capture some new contemporary samples, collect samples near some of the areas that we think are most impacted. And we're going to be doing this total fluorine measurement to get the totality. So if those concentrations are much higher than we're measuring in the target assay, obviously we have more work to do to figure out what these things are. And so we're always looking for volunteers for sampling and also funding as well. The other thing we're doing is once we've established what the total fluorine is, and let's say it's a big gap between what we're measuring with our targeted workflows, we use something called non-targeted analysis. And so what, I won't get into the chemical, the workflow, but basically what we can do is we can identify all these unknown signatures. So let's say we ran a sample and all these points represent a different compound, and they don't have to be PFAS. It could be all kinds of things, right? We have a software that we've developed, a chemical workflow that allows us to take that and pick out all those things that have PFAS signatures. And then we can go through and identify what all the unknowns are. So we can start to try to catch up to that total fluorine measurement. And that's something we're also doing in Tampa Bay. But one of the issues we've been running into is that a lot of these new PFAS may be low concentration. So we did a study, or working on a study right now, looking at collecting coastal foam, which is basically the stuff you see on the side of coastlines. And because PFAS themselves are surfactants, they accumulate in foam. And so a lot of our early data shows that these foam samples are between 30 to 1,000 times higher concentration in comparison to surface water. And some of the highest levels were in 100 parts per billion. So what we're excited about with this foam is that it will allow us to do non-targeted analysis, and we can actually look at a lot of things that we might not see because of the concentration in small little surface water sample grabs. And so we can actually get to some of those signatures, and those signatures are important because that's how we can actually start tracking where these PFAS are potentially coming from. We also are in the middle of a big comprehensive fish profiling project. To date, we have close to 2,000 fish mussel samples across 50 species, working with a lot of entities, including charter boats and seafood markets. If you have anybody interested in helping us collect samples, please let us know. And we also have a parallel freshwater fish project going as well. And just the last two slides quickly, we're also in the middle of a Florida-wide composting project. So if any of you do your own composting, please reach out to us. We're looking for samples tracking PFAS in compost. I'd be happy to discuss more about this another time if anybody's interested. We also are interested in tracking PFAS in unique wildlife models. We published a paper looking at PFAS and aquatic vegetation in addition to seagrass, and we found high levels in the Tampa Bay area. And then we're also looking for new sentinels. We have a project right now looking at using sand dollars because of their burrowing nature and sediment as a new sentinel for assessing sediment burdens of PFAS. And as you can imagine, the sand dollar is highest to things like MacDill Air Force Base, reflected what we found in the sand dollars. And lastly, I just want to point out, we're really interested in trying to start a project looking at PFAS in blood. There's not much blood data for Floridians. Blood is actually the main matrix to look for PFAS because PFAS actually bind to proteins in the blood, so they bioaccumulate there. And so looking at blood, we only need like very small quantities of blood, 100 microliters to do testing. And they've actually came out with these really cool sampling devices that you can actually send to people. They use a Lancet to have a little drop of blood. They put it on the tips of these Mitra sampling devices, and then they send them back to labs, and we can actually test PFAS on those samples. So if anybody's interested in trying to help start a study looking at vulnerable communities like the site Tampa Bay, please reach out. And that's what I'll have for my presentation today. Happy to answer any questions. If anybody wants to talk or chat about anything I've discussed or wants publications, just reach out. My email is on the bottom there. Thank you.
Just a couple of questions. Many questions in my head, but I'll just list a couple. Your heat maps look like anecdotally they would fit well with population maps for Florida. So I'm wondering if you've explored that layer, and obviously the firefighting foam is certainly a known source, and I would think you would have correlation with population and that as far as urban fires type of correlation. But are there any other kind of population concentration linkages you're exploring?
Yeah, so as you can imagine, it's a very extensive data set. So first part was just to get this paper out, and we've got this paper published maybe like a couple months ago. Now we're in that process of kind of peeling back the layers of the different ways we can use the data to kind of find those associations. So it is among the things that we've looked into. We've made some maps that show population density, and they do also reflect a lot of the same patterns we see in the heat maps, but we haven't done any statistical comparisons or anything yet. It's been mostly just visual so far, but we hope to get into that. And just interestingly, the AFFF usage, we mapped all the military sites in Florida, and I was surprised at how little they showed on the map. I mean, so I think for those places that have a military base, I think that is an immediate concern, but it doesn't explain the high levels we see across, the bulk of the rest of the state. So I think there's, a lot of different sources. And of course, PFAS have been used since the 1940s. So you also have a historical long-term use that kind of complicates some of this as well.
Well, that was my next question was, you've got the military application of that, and, the first thought is these foams are pretty gnarly and maybe they could be phased out. But with the increase in EVs and lithium batteries and how those don't play well with water as far as a firefighting mechanism, I'm wondering if these foams or similar are going to be actually increased in use throughout the US, not just military installations. And then how do you fix it? How do you get a foam that fights those fires that isn't so bad?
Yeah, the two things I'll say about that is one, they are trying to phase out the use of HFLFs. Although many installations have stockpiles of these, so they're kind of grandfathered in. There has been a lot of effort by the EPA to come up with non-fluorinated alternative foams. But unfortunately, everything I've seen so far, those alternatives are actually more toxic than the PFAS. So I don't know that there's been enough progress to kind of fix the solution. Of course, we need something, right? So it's kind of a hard situation because those who are putting out fires need something to be safe. So it's definitely an area of interest that a lot of people are exploring for sure.
May I ask a question?
Sure.
John, thanks for the great talk. Very interesting, and PFAS hotspot so scary. Most of them will end up at seawater, Tampa Bay, or coastal water. Finally, so a question for you is there a more portable or tool to test, to detect, or to monitor these PFAS in the seawater? Because you don't want to go to sea all the time with so many boat time. That's not affordable. So is there a sensor like our water quality sensor or other temperature salinity sensor, simple thing to detect that?
Yeah, so good question. So by trade, I'm a chemist, so I tried to get into out on the boat as many times as I can. So I try to get out of the lab. So to me, it's okay. But I know you're saying there have been some people exploring the use of handheld detection devices for PFAS, but I think they're mostly focusing on drinking water. At this time, I don't think surface water is a little more complicated for measuring, just because of all the potential things that are in the water, right? When we take water samples back to the lab, we actually have to do a lot of sample preparation to those to get rid of a lot of the background. So I think that's maybe something that people are trying to do, but there's definitely not anything available yet. And also, too, the other issue is that PFAS are very diverse in their chemical structure. So, those devices that come out might be good for a couple of them, but as we keep uncovering new types of chemistry, it's really just going to come down to doing them by mass spectrometry. And that's why the crowdsourcing techniques have been so great, because if we connect with charter boats and people who go out on their boat all the time, people who live in the area, we get them bottles. When they go out, they collect us a sample. And so, we don't have to be on the boat all the time. It's kind of getting everybody involved. And what's great about that too is then, we have conversations about the data and what we're doing. And so people get educated and people are aware of these issues. And so, when it comes time to get funding from the government or get people aware of, hey, we need to start cracking down on those that are polluting our waters, and the people know what they're talking about.
Yeah, that's great. Though I heard from you that the PFAS has a long more stable. It won't decay quickly. So it can be treated as a pastry tracer in seawater. So that reminds me of something we could help you to track PFAS in seawater. We have a very high resolution for Tampa Bay region numerical model, ocean circulation model. We applied that nicely to a Piney Point event. And also coastal ocean circulation model for the entire Florida coast right now. So we could track your PFAS in coastal water by using our ocean circulation model, nowcast and forecast. Yeah, that's something which we should think about.
Definitely. And actually, we actually wrote a grant recently, didn't get funded, but the idea we had was that when we go out and do our sampling, we go out with I go out when I think of where I believe PFAS are going to be based on sources, but that doesn't necessarily reflect where PFAS end up, right? We don't know where the residence times are. We don't know where they end up accumulating. And so we wrote a grant that I'd be happy to share with you. We wanted to come up with models. So if PFAS are released from a certain area.
Here we go. We should collaborate on this. That would be a perfect project.
If you can come up with a model using things like current and temperature and all the different things that they're plugging into the model based on all the data that's been collected for decades, right? If we can use that to figure out where these PFAS are likely to end up, then we can actually sample those locations. And those are data-driven locations that might tell us about what's happening. So maybe places we're sampling aren't actually the places that are going to tell us about, PFAS influx. The other thing I'll just mention too is that if you're modeling a lot of the known PFAS, you're not really going to get much information because, they're from so many different sources and they're coming in there from forever, right? So using some of this non-targeted stuff, we're actually monitoring some of these newer ones that are maybe more specific to certain applications. That's where we're going to be able to do some modeling and actually track it back to the point sources. So I think definitely let's talk, because I think there's some really cool things we can do. And then as climate change continues to become a thing, you got hurricanes and other stuff, having models to predict, where those would go in the future, I think would be kind of a cool thing to do too.
That's what we do.
Yeah.
Yeah, thank you.
Yep.
So I have a comment and a question. First of all, I just want to commend you for putting the data available on your website and making that available to others that have interest. I think transparency is a really important part of this process of shedding light and building trust on these important issues. Something that we try to model here at the Estuary program to varying degrees of success. So I'd love to see other folks doing that as well. My question is related to Pinellas County, obviously, because that's where we're at and that's where we are most concerned among other counties in the area. You showed maps of PFAS concentration in drinking water as well as surface water. And what I noted from that is that surface water, Pinellas was the top in terms of number of hits above the EPA standard. But drinking water looked like we were doing pretty good. So what is that disconnect, especially because our drinking water comes from surface water for the most part? Is that sort of a testament to how well we're treating our water for drinking water, or something else?
Yeah, that's a good question. So of course, like with that drinking water project, we tried to network with a lot of the local water municipalities. As you might imagine, when you come knocking on the door saying we're going to do PFAS measurement, there's not really a lot of engagement by many of them, and so we wanted information about where they source their water, what some of the treatments they have, and so I don't really know exactly what each of the different water sources are in terms of the composition of whether or not they get from groundwater, river water, Tampa Bay water, or whatever there's the aquifer. And I don't really know what all the different treatments they're doing at each one of these locations. Interestingly enough, what the biggest thing that came from the project that I learned was that those that did talk to us, those water municipalities, they shared their data. So some of them are doing their own testing. And so if you're in a place that's doing their own testing and actually monitoring, now they may not be sharing that data, but if they're doing their own testing that's a good sign, right? That they're aware of it and they're trying to get ahead, because these are things that are coming from the EPA. But one thing we learned from those that did want to kind of talk to us was that our numbers looked different in some cases. And so what that really got us to start thinking is maybe there's stuff that's contributing between the water source municipality and our houses. So you have sealants on pipes and you have different materials that are being used to bring the water into the houses. So that's another thing that we're starting to explore, to look at, not just trying to keep engaging with those, that community, but also trying to see that maybe there's other things that we might be doing in our houses or on the way to our house that are contributing. So everything we do, we have, like you might imagine, tons of follow-up questions where we really want to understand what we're seeing. And so it's great that you guys are looking good now. But as you know, with hurricanes and other types of things that happen in Florida, I think it's still very important to be diligent and keep testing. So we're hoping to keep doing drinking water testing moving forward. We're going to keep putting all the new data as we keep getting it online. So we keep updating it. And again, be more proactive about this issue than not.
Yeah, appreciate that. And you made a lot of calls for volunteers for various projects. And if there's any way that our program can help disseminate that information, send it my way, and we could, share it through our socials or our various email lists. We're happy to do that.
Great. Yeah, I'm a chemist by trade, so I'm not really that savvy with the dissemination side. We're my students kind of help with that a lot, but absolutely. I mean, we want this stuff to be at the forefront, because the more people are educated about it, the more people want to volunteer and be active participants. And that allows us to do more sampling and get to, and also, local knowledge too. I mean, we sample across the state. You guys know more about your issues in your environment than I do. And so that information that we get from those people are extremely valuable to understand what we're seeing. So it's great two-way street.
Absolutely. Two questions in the chat also related to sampling. One, how can we get a blood sampling kit?
Okay. Well, right now, the biggest issue is it's trying to make sure we have the proper protocols in place to have people collect and do blood testing. So if there's enough interest and we have enough people that want to participate, we just need to go through and establish those protocols so that there's a safe connection of data transfer between us and the individuals. But I would say the first thing I would do is, people are interested, to reach out to my email, and I'd be happy to chat more about that. And then once those things become online and we're actually doing those, we will reach out and send those kits. I'll just say right now, everybody's going to have PFAS in them. So just as a preface, it's just how much you have. That's the key. So.
I mean, I get so optimistic when I hear PFAS presentations. It's like, it's got to be.
I'm not very fun at parties, unfortunately. People ask me what I do, so.
And second question online, shouldn't the sea foam be collected in a non-plastic container?
Yeah, so we've actually tested. We spend more time than I would like to admit testing everything that we touch and making sure it's PFAS free. When we started our lab, we spent the first 6 months going through every single item from collection to analysis. So we are extremely diligent about that. It's very difficult to collect foam. I would recommend anybody who's interested to seeing how easy it is to collect foam to try to do it. It's very challenging. Those Ziploc bags that we use are actually PFAS free. So we've tested them every time we use them. But yeah, those are important things to pay attention to whenever you're sampling, to make sure that you don't have any PFAS in your background. And we run blanks, so we have we put water in those Ziplocs, and that we know it's PFAS free. We take it through the entire workflow. So our projects are funded by the government, the EPA, CERTIP, DoD. So we have strict protocols that we follow to apply to everything we do that make sure that our backgrounds are PFAS free. But good question.
Excellent. Any more questions for John? I have more, but I'll let you go.
Yeah. Well, I mean, I'm here for you. So I'm probably gonna go back to the conference when you're done. But if you have any more questions, let me know. And if not, you can always reach out via email.
Yeah, can you actually drop your email in the chat?
Sure.
Yeah, and sincerely appreciate you being here. Like I said, you know, you're at a conference, so I know this is taking away some time from that, but appreciate you being online, and there's a lot of interest in this topic. And yeah, if there's any other questions, you can follow up with John. And John, like I said, you can contact us if there's any way that we can help distribute information about College for Volunteers.
Great. Yeah. And if anybody wants any of the papers or any of the publications, you know, feel free to reach out to you. I'll be happy to share those with you. All right. I'm gonna see if I can figure out how to get back to the chat. But thank you for the opportunity.
Okay. So our next presentation is actually going to be a recorded talk from Amanda Mooney Morgan and Jay Law. Going to be talking about the Tampa Bay Observing Network. And they were so gracious in providing this recorded talk. They've been really inundated with post-storm cleanup and work to make sure their network's back online. So they're actually in the field right now on a boat, calling in through their phone. They are online only to answer questions, but for the sake of preserving bandwidth, they thought it was better to send a recorded talk. So that's why we're doing that today. So I will get that started. And the recording is a little bit quiet. So everyone just please be extra quiet in the room while this is playing. We should be able to hear it over the speakers, but we'll try our best. Amanda, Jay, can you verify that you can hear us? See, you're trying to talk, Amanda, maybe. Either way, they are online. I'll check during the presentation, but let's start the talk.
Here to be any sound.
We have a pretty big group at the Ocean Circulation Lab. Our directors are Dr. Youngin Liu, and our founder is Dr. Paul Weisberg, who is emeritus faculty. And it ranges from scientific staff, so postdocs like myself, technical staff, which is really important and vital to the work that we do, undergraduate interns who are essential to all of our operations, also graduate students. And it's our expertise range from modeling all the way to water quality and engineering. So it's really a diverse group that allows us to have good interdisciplinary work. So we're going to check the resolution of the distribution model, or putting these on one of those. Some changes to the machine. So first,
Sorry about that. So Young Gang has volunteered to give a presentation on his end briefly over probably about 10 minutes. He's got I think a defense that he's got to head to, but I will give him controls. Let's see. And he will take it from his end.
Let me
should
see where I'm gonna share my screen. Okay. So you see my screen now?
Yes.
Okay. Yeah, sorry for the inconvenience to everybody. This yeah, on my screen there is a follow one allow for 1 month the open system. There's a do you see the little warning sign in the center of my screen, or just totally fine for you for people?
Yeah, we see that.
So how can I remove that? Stop now.
I would just allow it for 1 month, maybe.
Open setting. Oh, it goes away. Okay, right? No. Okay. Okay,
There you
now.
go.
Yeah, thank you. I only have 10 minutes also because the student defense to start at 11. So I could go briefly. Not everything Amanda has talked. Okay, Amanda is a postdoc in our lab. Jay is a lead in seagoing activity. He is the main powerhouse behind this T-bond work. So I serve as the lab director of this ocean circulation lab. Also, I'm the PI, lead PI of the T-bond project. Okay, so I will go to the main point. The T-bond project started as a result of Piney Point wastewater emergency response funded by FDEP. Back then, we were able to use our high resolution coastal ocean circulation model called TBCOM to monitor this wastewater dispersion and tracking the polluted water in Tampa Bay and provide guidance for water level, wastewater sample collection. And this result has already been published in a peer-reviewed journal, Marine Pollution Bulletin early this year. Interested people, you can take a look at that. This work led to the T-bond observation, because back then there's no observation, continuous time series observations at the time. We were all just discrete in time and space water sample collection. So nicely, FDP found this project. So we were able to put a total of 5 water quality observing stations in Tampa Bay, 4 in the middle of Tampa Bay, 1 at Coastal Water College Marine Science, very close to our lab. So we call TP5. So TP1, TP2, TP3, TP4, TP5. We have another one, TP06 is planning. So we put these water quality sensors in the U.S. coastal range towers. So see this we provide a plot, a figure shows this the tube. That's where the water quality sensor is housed within that tubes. We also have full meteorological data sets or delivered through the system from surface. These are the cool pictures we taken from the after recovery of the sensors. You can see a lot of biofouling. That's a challenge for observations, water quality observation in summer, particularly in summer. So it's a learning process. We learn a lot. So how frequent we're gonna maintain all these? Okay, so Amanda play a major role in data quality QA/QC of our data. She was hired here. I think that's the main purpose. My assignment to her to do this, and she did a great job. And she's now also try to implement this QA/QC in real-time data. Real-time data served through our website we call T-bond. Not only the plots, but also data you can download, you can access. So this is a typical example, shows that the data we collected during the Hurricane Debby, and back in in early August. See that? Nicely, this after this hurricane event, we see the temperature drop quickly, and we see that particularly we see the chlorophyll slowly may increase concentration after this event. That's in response to this Debbie. Debbie caused some flooding in Tampa Bay region. That's some coastal water being flooded into the bay after during that event. Of course, not as serious as Hurricane Helene, but back then it was the coast issue. Sarasota was flooded particularly very badly. Yes. Anyway, we see Tampa Bay the water response to that hurricane event recorded by our T-bond sensors. So for future plan, Tampa Bay Estuary program contact us. They could use this Honey Point penalty money to start a new site at Bishop Harbor. We call TB07. Maybe some other names in the future. We will discuss about that. So we're trying to expand this water quality observation effort within Tampa Bay. Yeah, expansion continued, not only here, but also to the south. We also plan to expand further to the upper Tampa Bay. We could TB08 in Hillsborough Bay, and in Old Tampa Bay is a TB09. We try to seek funding for this. So we because this is really what I talked will not be the exactly Amanda wanted to talk, but we had the discussion with FDEP and also Tampa Bay Estuary program people will add the power sensors to our TB-bond network, and we could mirror more and have more information. Yeah, that's I think on the break, so that we can have the main information out there. You guys know what we have, what we're gonna do in the future. Yeah, we are actually seeking funding for this, and in collaboration with local management, like Tampa Bay Estuary programs. So I think Amanda and Jay can take questions. This is much better than just to play a very low, no sound video, poorly recorded video. Yeah. Okay. I'll stop here. Less than 10 minutes. Probably you guys get this information in a shorter time, save time for everybody. Yeah.
Yeah. And can you guys hear me?
Yes.
Yeah. Thanks for jumping in there, Young Gang. Yeah, I'm not sure what happened there. We tested out the audio before we left on Monday afternoon to go out in the Gulf, but we actually are connected through my iPhone through satellite Wi-Fi on the boat, so I think we can hear most of the questions. Some of the audio we're having a hard time picking up, so we might have to have any questions from inside the room. We might have to speak into the microphone, but we can easily back up if you want to ask questions about some of Amanda's work. Yeah, sorry about the complications there, but it looks like we're rolling with it. So it's been a crazy, obviously for everybody, a crazy few weeks. We had quick backstory. We were supposed to be out on the Weatherbird servicing our offshore moorings before Helene hit. We had to postpone. We postponed it 2 weeks. Then Milton hit, and we're out. We were out Monday and Tuesday and today to get this work done. It's the only window we had through like November. So I think we just tried to do everything, and we're getting bit with some technical issues. So I'll hand over to Amanda. If you guys have any questions, we can just start the discussion, and I guess just kind of free flow.
I'll stop sharing to take questions.
Questions for Amanda, Jay, or Young Gang?
I have a question, just kind of related to the storms that we just saw. Do you guys have data through Helene and Milton, and any plans on kind of a short response summary of what the bay waters, what you saw at your stations through those storms?
Yeah, great question. And that was on one of my slides, and hopefully you guys can download that PowerPoint. Our system has proven to be robust through Hurricanes Debbie, Helene. And right now we're feeling our sites on the Weatherbird for Milton. We do have data for both Debbie and Helene. Helene, we had some data go offline because our servers went down at USF, but that has been backfilled. And we also have the internal data. Debbie's being worked up right now for a manuscript. Ultimately, we would like to compare hurricane events because they had different effects, you know, rainfall effects, storm surge effects, etc. But yeah, Milton's to be determined. Do you want to speak on Milton at all?
Yeah, so as Amanda said, the system was really robust, and we know that Debbie, Helene, and Milton were three very different storms. So the surge from Debbie and Helene, we picked that up. We did see a response in the chlorophyll. The system measured throughout both those storms. The online data went down briefly through Helene because we lost power at the USF. Milton is a much different story. The high winds really caused some issues. Actually, on the way out on Monday, we were able to go by one of our stations. We have 4 stations that are on Coast Guard range towers in the bay, which make it for a really nice platform for bolting instruments to, the telemetry, the power systems, and everything. One of those towers was actually blown over in Milton, which we didn't know until we were on the way out of the bay on Monday. So that station is actually gone. But you know, that's what happens, I think, with you know, 100-year storm here with 100 plus mile an hour winds. Actually, we're on the way in the bay right now, and we're going to be hitting the other 3 stations that are on range towers just to assess the damage. But really, Milton was really historic. We were out servicing our offshore moorings. We have a mid-ocean buoy off of Sarasota, and then about 20 miles off Sarasota, and one 60 miles off of Tampa Bay that we serviced just the last 2 days. The one off of Sarasota was completely destroyed. We've never seen that with any hurricane. We've had hurricanes hit them before. But everything on the surface of the buoy was just gone. So yeah, I would reiterate that I think the system is really robust, but when you get a storm like Milton, it's just, it was pretty devastating. But we are working up the Debbie data, and the Helene data will follow. We definitely saw a response from Debbie, like 2 to 3 days afterwards. The chlorophyll started to spike up at all the stations. Yeah.
Thanks, Young Gang.
Thank you. Yeah, I need to run now.
Bye.
Tell Alex good luck.
Oh.
Yeah, and just to reiterate, I guess what was in the last slide, you know, this is a relatively new effort, and finding continued resources to support it, I know, has been kind of a challenge. So any means to, you know, develop some collaboration with this group to support them, or collaborative grant efforts is definitely something that we're thinking about. As a case study, I guess the new stations at Old Tampa Bay, Hillsborough Bay, and the one in Bishop Harbor were supported through litigation funds through the Piney Point settlement. So that's, you know, a case for the power of litigation to make things happen. But you know, sustained funding to support this is definitely, I know, on the radar for this group. So like I said, you know, any way that we can figure out a way to support this and you know, perpetuity, that's what we're looking for. And we've talked about continuous monitoring data in Tampa Bay for a long time. I'm personally very excited to see this running and see the data streams coming in. I'll make use of it, but I know there's many other applications that can come out of it as we're explaining. So I think the future's looking up, but it's just more of a question of making this sustain in the future.
Yeah, thank you. Sorry, I think we're picking up most of that, but yeah, I just wanted to follow up on that. That these stations, they're in Middle and Lower Tampa Bay because this came out of the Piney Point response, and we are aware that, you know, there are much more impacted areas of the bay. So that the greater plan is to expand to Bishop Harbor. Like I just said, I'm not sure if that was Marcus or Ed. Sorry, but yeah, so that one will definitely be going in with support from Estuary program. So collaboratively, we'll be doing that, and we are working on a proposal with through the EPA right now to add a station in Hillsborough Bay and then up in Old Tampa Bay, so that we can start really to dig into those, you know, those areas that are more impacted with longer residence times. So yeah, we're pushing forward.
I think the EPA proposal will be very competitive.
Yeah, I think so as
Thanks,
well.
we hope so.
Cross your
Yeah,
fingers.
Go ahead, Marcus.
I'm just saying, cross your fingers for more funding.
Yeah, absolutely. And now it's, I mean, 3 years, that's nothing to laugh at, for sure.
Absolutely.
Yeah, and I would also add that FDEP has been really supportive of this project, and they actually reached out right after Milton, and I believe they do have some possible damage funding. So that's part of the trip out here this week, was to assess what's going on and just to get a handle on, what kind of damage we incurred and what that's going to look like to get things back up online.
All right. Well, I don't see any more questions online. No more from the room. So I think you're off the hook. Again, we really appreciate you guys trying to make this happen amidst the circumstances. Thanks for jumping online. And yes, the recording for that is available on the agenda. That is linked in the meeting invite for this. So you can download that and listen to that on your own. But yeah, thanks to Amanda, Jay, and Young Gang for that. We'll be talking.
Thank you, guys.
Thank you, everyone. And one last thing would be, one of our stations is at the College of Marine Science at USF. And if anybody wants to learn more about this, just reach out. You can come down to our facility and we can literally look at it. We can talk you through what we're doing with the water quality observations, the Met observations, and all the real-time systems. So thank you.
All right, field trip. Okay. Last presentation we have today is from Ray, who will be talking about, not about Milton or Helene, which is, I don't think he wants to talk about it. They'll be talking about some updated modeling efforts. What have I done? For historically mined lands in the Alafaya watershed. And I'll pull up your presentation.
The memory just split second, so you get the title out. Well done.
There we go. It's my job to run this.
That's where you get the big bucks.
Whatever you want to call it.
Yeah. Okay. We had done some work at ESA. I wasn't involved at the time. This started back 4 years ago or so. And Doug Robeson and Ryan Horseman and a couple other folks, Bob Worth was involved. I've been doing some work with Tampa Bay Water. Let me just run through a little bit of what was going on there. Tampa Bay Water had gotten a grant through the to do this study, which had been identified as something that was needful in the Habitat Master Plan from 2020. And Tampa Bay Water has source water assessment protection program effort that Bob McConnell, bless his soul, was working up, and kind of a testament to his vision that we will continue to be working on this stuff for them for decades, I'm sure. So ESA and our subconsultant WSP was looking at real observed flow data and water quality data coming out of historically mined lands in this was actually the south prong of the Alafaya River Basin along the Alafaya River State Park. So they had some measured data. And the reason we were interested, and I got pulled into it, was a question of how these measured data compared to what we have been doing for years with our model-based loading estimates for not just the Alafaya River gauged areas and ungauged areas, but all Tampa Bay watershed, but special interest in the Alafaya River because there's so much of preponderance of these previously mined disturbed lands. You can see that all that yellowish and oranges is stuff that's been impacted by mining over the last 100 years. So 2/3 of the watershed itself. So Doug asked me, hey, do a comparison. We've got these observed data down at this little creek here off the South Prong. I think this measures about, I think it was 170 acres or something. So 700 acres. This little bitty piece here at McMullen Branch had been previously mined. This was part of an area where the stream bed was more left intact, and had gone out and collected samples over I think a 10-month period or so. Extrapolated it to annual loadings. And now we wanted to see how that would compare to what our estimates from the model that we've been using to estimate loadings in Tampa Bay for the last 30 years, how that would compare, just to see how different or how similar, but what it can tell us about how we may be needing to revise our model to better account for these previously mined lands. The model as we use it is based on land use specific characteristics from the district land use. The district land use, this little yellowed watershed may contain upland forest and some wetlands and some streams, but it's not identified specifically as a previously historically mined parcel. So the set of coefficients we use in our empirical model to derive some type of loading estimate from that is dependent on a set of parameters that are related to specific land uses, but those land uses are not identified as being part of a previously mined area. So we had the data from WSP and their estimates of what the average loadings were for the year. Specifically, we're most interested in T and TP. Those are two of the things that we simulate and measure as well for the Alafai River at the gauges. We have gauged flows and gauged and measured water quality. We can estimate loads at the USGS gauges at Lithia and at South Prong and North Prong. But in this very small little watershed, we also have these estimates, and we can compare those to what our model might say and get an idea of how different things might be. The unit area stream flow, one of the biggest points here is this third bullet here. The unit area stream flow was considerably less than what was expected. Had it not been mined, we would expect a more familiar runoff pattern. But since it had been mined, as we sat around and brainstormed about what might be going on here, Doug Robeson, especially with his, I don't even say how many decades of experience running around through these different watersheds, it just makes sense. This thing has been torn up, laid back down, restored, in quote marks. And the seepage and drainage characteristics in these pieces of the watershed are very, very different than it would have been had they never been mined. We also, as part of that WSP work they did on this, they did find that stream flow was a lot greater, totally outweighed any type of seepage flow that might be coming out of the system through groundwater. So the only thing we're really concerned about here are loads from the stream, the surface water flows themselves, which was an important finding when we're looking at what's going on throughout this entire, 2/3 of the watershed. So we had our non-point source loads that we had calculated based on an empirical model that had run through 2021. And we could compare them to the data that came from this small little point down here. This little hatched orange area is a sub-basin. About a third of that, or 40% of that, is made up of this little McMullen Branch watershed. So we had an estimate coming out of the sub-basin and take our 40% and compare to the measured data, and all's hold, we can split the entire Alafai watershed up into these three basic basins. Everything in that light blue, when we do an actual loading estimate that we report to all the partners, is based on observed data, not our model-based data, but observed data at the Alafai River at Lithia Gauge and the EPC long-term water quality monitoring sites. So those data are the best available and most representative of what's really coming out of the North Prong and South Prong there at Lithia. But for this modeling effort, those observed data is not what we use to compare to our McMullen branch data. It's a little model that we had developed 30 years ago to use to compare. So for McMullen branch, the nitrogen concentration as measured was about 0.7 milligrams per liter. Using the land uses from the district land uses from, I think it was 2017 at the time, and coming up with an average concentration of what nitrogen should be for that 700 acres. Our nitrogen concentrations for a model a little bit higher by almost 40%. For phosphorus, it was just exactly the opposite. The observed phosphorus data coming out of there were 2.5 times as great as what our model said those concentrations should be. So obviously there are some serious differences here. We did look at 3 different years in the most recent 5-year period considered to be a normal year, a wet year, and a dry year, but the actual differences between the model and the observed data during those time periods didn't change all that much. So we're just going to report what we found for our one normal year in comparison to what the observations said. And if we extrapolate the data from this one little watershed to the entire Alafia River watershed mined lands, that 2/3 of the basin, then our model says that about 30% of the total nitrogen load from the Alafia River watershed was from mined lands. We use these observed data from that one watershed, we only get about a quarter for nitrogen. For phosphorus, our model said it's about 30% came from those mined lands. From this McMullen branch data, it's closer to half of the phosphorus coming down the Alafia may be from those mined lands. So it gives us some room for thought about just how important it is to get this information right when it comes time to getting an estimate of how much phosphorus and nitrogen is actually coming off of these disturbed and refurbished mined lands. We did some quick checks and just compare for that normal year. We can run through this if you'd like to, but it says the same thing. Basically, the nitrogen loads from a model are somewhat more than what the observed data say, and the phosphorus loads from a model are somewhat less than what the observed data would estimate. We also did a quick comparison of the hydrologic load that was measured coming out of McMullen branch and compared that to what our model would say, and the measured hydrologic load was only about half of what our model said would be coming out in the absence of it being previously mined, which matches up with what we've been thinking about these disturbed lands allowing much more water to move down through the soils instead of running off through the surface water. Okay, so just a quick summary at the back end. So we were, based on this, if that all those mine lands in Alafia River watershed behave similarly to this McMullen branch 700-acre watershed, which was selected because it was considered to be representative of what those historically mined lands are in Alafia, and it was accessible because it's publicly owned lands. You didn't have to go on mosaic property somewhere to try to do some sampling and get all that cleared up. So, but if those historically mined lands act similar to the McMullen branch, then if we start looking at model-based estimates of loads coming out of Alafia, we're going to have to consider potentially revising our coefficients for runoff and for nutrients coming out of those portions of the watershed that have been mined before. And we typically use the model-based that far up in the watershed if we're looking at, say, potential future loadings coming from the situation. If we extrapolations of what mining may occur, or urbanization may occur, or even climate change may occur, we're doing future assessments of what we expect to happen. We do use model-based estimates. So this gives us some thought. The next time we run around and do that, we need to keep this in mind and perhaps change part of our parameterization of our model. So please ask questions. There's a whole lot more that went into this whole thing with this project from Tampa Bay Water than what I just talked about, but this is a little part of the loading that we thought would especially interest the consortium, you folks, and anybody else involved with the whole loading thing we've been doing for 30 years. Yes.
So, you're saying that this 723 acre McMullen tract was reclaimed. So this is officially reclaimed and let go by the FDEP
That's correct.
and considered to be more or less normal.
As normal as they can get, yeah, right. And as part of that Alpha State Park, you actually go there and walk around it if you'd like to. So you know, visually, some of the photos look like it may well be what you expect out in those seasonally inundated. You have a nice little stream during the dry season and may have some base flow. But if you put a shovel down, dig it up, you get some odd looking things compared to what you would normally expect.
Yeah, so that was one of my questions. I mean, historically mined, how does that literally change the land? I mean, it's a change in topography, change in, like you said, how the water infiltrates.
Yeah, well,
all that stuff.
so there's the two classifications based on the temporal period in which the mining was done, mandatory and non-mandatory, one's pre-75, one's post-75. I think I included something about that in here somewhere. So yeah, so pre-75 is called non-mandatory mined lands. Most of those in the Alpine Basin are, well, they look something like this. They'll be backed up. Almost all the drainage will be internal, very little off-site drainage. And you can see that when you even just drive, you know, the day of the roads through here, you see that. Those are the pre-1975. Post-75, rules, regulations required that mining companies try to restore as much as possible the natural hydrography and ecosystem that was there before they ripped off the cover and took the phosphate matrix out. So that's the difference. I mean, that's why you see this mix here. Obviously, that's a whole bunch of it's mandatory along that eastern side, and then some non-mandatory, but looks like it's more realistic streams, and the main channel of the Alpha, which thankfully was remained untouched for the most part. Yeah, so that's the difference. So when we do our actual the loads that come out at Lithia, at the Lithia gauge, the USGS Lithia gauge, any rain that falls on these old pre-75 mined lands is more than likely staying there and not draining out the river, out the South Prong into the main channel. Unless we get a very large event where some of these dikes get overtopped.
With the difference between the N and P kind of showing up the model versus observed, to your point, those pre-75 drains internally, but that would be both N and P would be staying there. I'm just curious, in my mind, I can see the P, and this is not the right terminology because of my ignorance, but being mobilized a little bit more, that's what they were after, right? You know, that's what they're mining. So in my mind, conceptually, that becomes available. This would be the post 75 where it is draining into the basin. Do you have a concept of why the N would be reduced in that scenario?
No, I don't.
Yeah.
I don't know why. We're gonna have to. It will be interesting to see how far down the path of how important that is and why that's happening, and
yeah,
who we get to say, okay, we got to figure this out.
Yeah,
I would like to know. It's great, so. Yeah, we'll be interested in that. It would also be interesting to know what you know. We have a model, but it would be interesting to know in real life. You know, before the Alpha got hit with mining companies taking this Iowa rock out. How was the flow? How did hydrograph look at Lithia? How much water is actually being retained up in there? I mean, we can make some estimates, but that's got to have some kind of impact on the actual flows and loads that are making it down to Tampa Bay.
Yeah.
But we haven't done that. I would like to, but it's just play with it, you know, in my free time.
Well, I guess you would just taking it back to the hydrology, if you have less than assumed, less water than model, your model would show going in, maybe that's the limiting factor of in, and the P is just somehow exacerbated because of the process.
Yeah, it could well be, and then, I mean, it's the outlier.
Yeah.
Phosphorus, more phosphorus and shakes to get.
Yeah.
And for the most part, you know, for what we do as far as loads in Tampa Bay and how it impacts the ecosystem, that's phosphorus isn't ever limiting anyway. So, you know, it doesn't concern us as far as management goes. But yeah, as people who think about this stuff, we're curious as to wonder why that is exactly. You know, I'm not a chemist or a geologist either, but yeah, I would like fun.
Are you saying then that these mine lands are acting as a significant recharge then to the aquifer since the water's not running up?
They are taking water now. Where's it going exactly? I don't know. You know, I don't know how deep, what level it might be going to. I don't know that. But yeah, it's not draining off in surface waters. So we do know that. It's not coming out like we thought it should. Which is, it's not surprising. Yeah, go ahead.
I'm just curious because of the differences on what you expected
Mhm.
based on, you know, the water inputs. Has that led to any conversations internally about, you know, how these lands should be restored, either the same or different ways than they have?
No, they've been talking about that for decades.
Yeah.
You know, I remember years and years ago when I was fresh out of grad school and didn't know anything. I got assigned to go out by some law firm. I got assigned to go out in the field with folks who were actually sampling reclaimed lands. Just as some part of some lawsuit. I didn't know anything, but I did know when I saw, you know, rusty water running out of the ground, just in bubbles, that wasn't right. So yeah, we're still trying to get it right. And it's never gonna be perfect. But to be fair, places like that, like this little creek, they're amazingly productive. You know, if you didn't know any better, you go in there, you still got your ecosystem that's functioning at some level. So it's not like it's, you know, poisoned with toxic metals, like some of the stuff was previously. You know, this diked in stuff. Who knows what's in there? Not as bad as what's in my house, close to lean, but it's bad stuff.
You're not allowed to talk about that. So I follow up, I guess, to Brandon's question, you know, so using the observed data, you found that N was lower and P was higher. And I guess this also relates to the consortium. How does this really affect the bottom line of the nitrogen loading estimates? Because NPS is the largest contributor for, I think, all the base areas, right? So does this really make a noticeable difference in our load calculation?
This will make no difference in our load calculation. Because this is a difference between something observed and what's modeled. For our estimates of loads, we use the observed data at Lithia gauge as the final downstream arbiter of what's real coming by there. It does make a difference, however, here we're going to get esoteric in the consortium will of this. It does make a difference in how we split up the loads above that gauge to each of the entities responsible within the watershed. Because that algorithm includes runoff coefficients and nitrogen land use specific nitrogen concentrations. That can make a difference. And what it means is, you know, if the nitrogen coming off of one of these types of parcels is less than what we thought it was, that means that everybody else that doesn't have that piece of non-mining, non-state, so Hillsborough County, somebody would actually be responsible for more load, which if you want to start opening up WQ bells means greater allocation for them. Not that anybody needs that right now in that basin, but yeah, there are all kinds of implications if we were to take it that far. I don't think it's necessary.
Yeah.
But yes, there are implications.
Big picture, you know.
Yeah, yeah.
It's all connected, and you're not retiring anytime soon, are you?
No, I wish I could, but I don't think that's gonna happen.
We'll be around for a little bit. All right, cool.
Yep, I'm not going anywhere. Y'all have my cell number.
And this was a Key Birth project, you said?
Yeah.
Yeah, and I think the final report is gonna be available very soon.
Yeah, if it's not already, I haven't seen a final report yet, so I know Doug was putting it together, it's supposed to be ready, but I was a rate limiting step on that, getting this stuff done for him, so don't point the finger at too much, but yeah, that should be in your hands anytime now.
Cool.
Yep.
More questions?
And if
All
something
right.
bugs you in the middle of the night, you wake up, think about it. Call me.
Appreciate it, Ray.
At the moment, yeah, that's not what I'm thinking about. It's worth it. Holy smoke.
So that's all we got for today. If there's anyone who wants to have an open discussion about anything, just wants to vent about post-storm recovery stress, that's fine too. Everyone's doing good. Everyone's still at like a 7 and above. After that, yeah, I was gonna say after that PFAS, I'm like 6.5 at best. Okay, well, just a couple of announcements. First, we should be getting the tentative dates for the 2025 TAC meetings out to you probably December. I think we send that email out. So just when that goes out, make sure it gets on your calendar. We're always, of course, always looking for presenters at the TAC. So if you have a burning talk or know somebody that you think would be good to bring to the attention of the group, we're always open to those suggestions. Other announcement I have is I haven't made this too vocal yet, but we are going to be having a hackathon in January 27th. That's something that I'm leading with a lot of help from people at TBEP to organize. But that's going to be at the Tampa River Center, an all-day event. I have never participated in a hackathon. I know Blake has, for example, but if you're not familiar with what that is, you're basically, I don't want to say locked in a room, but you're confined to a space for a certain period of time and basically working collaboratively to develop a tool or product. And we are funding this effort, or rather we're organizing it to be a competitive process where the winner of the hackathon has the opportunity to enter into contract with TBEP to fully develop the product or workflow that's created during this event. And the idea is that, you know, we as a program, we have a lot of things that we report on regarding status and trends, things that we've been tracking routinely over the years, like water quality, nectin, nitrogen loads, all sorts of things. And there are some things that we know we should be reporting on, but we don't, because we just haven't developed a workflow to do that. And it's my hope that people that come to this event will, you know, pick a data set that's ripe for developing a workflow to report on and sort of operationalizing that, using some of the open science principles that we care about at the Estuary program. So for that, the registration is not open for that yet. It's going to probably go online first week of November. November 4th is our date. You'll see email blasts for that, I'm sure. There's going to be a web page on our website that tells you all about that. But we're eager to see how it goes. Should be an interesting event regardless. But just keep that on your radar and look for that announcement. So that's all I have. Thanks to a good year of TAC presentations. Appreciate y'all. Again, those online, please consider coming in person. It's always a much better experience in person. And yeah, we'll see you next year. All right, thank you, everybody.
Hey Marcus, sorry about that PO miscommunication. We've got it underway. I was like, oh.
Yeah, no worries. I just have these notes on the calendar from like,
Very good.
whether it's timely or not.
Yeah, it's fine, thanks.
See you. You're really good luck.
Thank you, yeah. We're told about 2 months, we're gonna be under repairs.
That's not bad, actually, considering.
I think it's optimistic.
Yeah.
I don't know. I really appreciate some communication.
On my way and I was.
They're trying to figure it out. They're like, I think they're gonna test the windows. So we're gonna see if we can, I mean, I just came in like over the weekend. I'm like, oh, cool, they were so cool. Fantastic. I wish they would have told me because I would have. Yeah.
It's actually the ones I changed.
Trying to be really not.
Yeah.
You wanna say hi to Marcus? Yeah.
They said they might.
Yeah, a lot of days. Okay, we got a little bit of water in our garage. Faster.